Good morning, my name is Carola Gómez-Rodríguez, and today I'm going to tell you how we have been using distance-decay models to assess historical dispersal limitations. So just let me share my screen. Here we go. Okay. Well, today we will be talking a lot about distance-decay patterns, which you are probably familiar with, because it's kind of the first law of geography, but applied to biological communities, in the sense that it just means that two biotic assemblages, two communities, will be more similar the closer they are. And you will see a decrease in the similarity of biological communities. That means in the number of shared species and the number of species that they have in common, that with this spatial distance. This pattern is well known. It has been well studied, as we can see by the number of citations to the conceptual framework by Nicola and White. One of the reasons many people use it is because it is a useful quantitative description, and the fact that we can easily quantify this pattern with a slope or an intercept allows us to compare it between regions or between different taxa. So for those of you who have not studied this pattern before, I will explain it briefly. So what we mean with the distance decay of biotic similarity is that if we represent biological similarity against spatial distance, we will observe that it decays, like there is a trend, a decrease, because two localities that are close to each other will have more species in common than two localities that are farther apart, as we can see here in the case of locality S1 and S3. But as you can imagine, two localities that are close to each other not only have a species in common, they have many other things in common, like the habitat, the climate, other factors that might be also affecting the biological communities that we are studying. And this is why when we are using distance decay models, we have to be fully aware that there are different processes that might be driving this pattern. Basically those related to niche processes, like I'll look into this again, climatic distance, and also the ones that are more like geographic structure, spatial structure driven by dispersal processes. As I have said at the beginning, it's very useful because we can quantify it, and that means that we can estimate parameters from this pattern, like the slope, the rate at which assembly similarity decreases with distance, or the intercept, that is the similarity at the closest sampling localities. This is easy to see just with if we compare two distance decay patterns. In the blue one, we have a steep slope. We can see that when distances are quite long, spatial distance, they are quite long, the similarity is low, while this is not the case in the round one. And we have high community similarity at large distances. And if we think about dispersal processes, we can easily figure out or easily see that if those flatter slopes are more likely to be seen in a species that can disperse well, with a strong dispersal ability, because if two localities are farther apart and they are sharing the same species, we need the species to have been able to reach there, right? In the case of intercept, what we are talking about is this similarity at the closest distance. So when two localities that are very close, do they have similar species in common, or do they have the same species in common, or there is quite turnover even at the closest distances? So with these things in mind, what we have been doing, what we have done these years, is use the distance decay models and patterns to assess historical dispersal limitation in different taxa. what we mean with historical dispersal limitation is that not so long ago, but 20,000 years ago, the northern latitudes were covered by an ice sheet, and since then the taxa that were restricted, the ranges were restricted to the south of Europe or the south of North America. They had to recolonize those northern latitudes. So what we are trying to assess is to which extent they have been able to expand their range to those localities that now might have this suitable climate, suitable habitats, but they are still the way to get there. So we are interested in this recolonization lag. Why are we interested? Well, because it's interesting per se, but also it has a clear applied interest, and it is that we are all wondering what it is going to happen with biodiversity in the near future with this climate change, and we have a great natural experiment. So if we understand what has happened in the past, to which extent biodiversity or these species have been able to track their climate, we can make educated guess about how they will be able to cope with it is coming, right? So I'm talking about biodiversity, and we talk about biodiversity, we have to talk about biodiversity. We cannot forget invertebrates. We know much less about their dispersal abilities than we know about other taxa like vertebrates or even plants. Well, so they are a huge fraction of biodiversity. We know very little about their ability to cope with what it is coming, but they are also quite convenient for this type of studies. They have been for us because some invertebrate groups, like arthropods, have attribute morphological characteristics that can be related to their dispersal ability. So if we are able to find signatures in the biogeographic pattern that we attribute to this dispersal, we can relate to this dispersal or morphological traits related with this dispersal ability. If we identify this signature as the result of this incomplete recolonization and this historical dispersal limitation, then we can try to see if in other invertebrates or in other taxa in general, we see the same signatures, because those could be indicative of strong dispersal limitation. So today I'm going to talk about 3 studies, the distance-decay patterns of beetles in Europe, also how we have used distance-decay models in the context of phylogenetic scaling below the species level, and finally distance-decay, but distance-decay in this sense, in genetic structure, because in this case, even if it's the same, we are looking at an increase instead of a decrease. So in the case of Europe, we have observed that if you divide Europe into the territories that have been recolonized from the ones that have been mostly refugee or like North and South of Europe, in some particular beetle taxa, we had observed that the distance-decay patterns in the north were different from the ones in the south. So we were wondering if this could be like an evidence of this dispersal limitation in this taxa. We tested it with many beetle groups. Our hypotheses were that the distance-decay patterns in the north would be flatter as a result of incomplete postglacial recolonization, because the species in the north are the good dispersers. That also the north-south difference in the distance-decay pattern would be related to the north-south difference in the species richness, because they would be the result of the same process, this incomplete postglacial recolonization. And we also hoped that we could find stronger evidence that the difference in the distance-decay patterns were actually related to the dispersal ability of the taxon. So good dispersers would be the ones which would have more similar distance-decay patterns in the north and in the south. So basically we saw all that. We confirmed our hypothesis, but I will tell you, I will show them in more detail. So with these 21 different beetle taxa, we observed that the distance decay patterns in the north were flatter, but not only that, they were also quite similar. So the difference in the distance decay patterns came from the differences among taxa in the slope in the south. Okay, so the north flatter than the south, but also quite similar among groups. We also observed that this difference in the slopes between the north and the south was correlated with this richness gradient. So we were like quite in the track, or like this is going to be historical dispersal limitation, yes. And as you can see here, for carabus, which its wing legs, most species do not have wings, so it's strongly dispersal limited. The slopes are stronger. These are just two examples to show you how the patterns look like. So we also tested if this difference in the distance decay pattern was actually related with those traits. And yes, it was. We didn't only related with dispersal traits, also with other ecological characters, but it was the dispersal ones, the one that explained most of the variance. So we were like, hey. Just a side note is that when we say we compared the distance decay patterns in the north in the south, we did not only compare the slope, we computed this index that takes into account both the intercept and the slope, because you can have same slope with different intercept, and that is actually quite different distance decay pattern. So with this study, we saw that the distance decay pattern in the north were relatively flat. It's probably only the good dispersers have reached those latitudes so far. That the dispersal attributes of the taxon explain the difference in the distance decay patterns. So we think this is clear evidence that historical dispersal limitation is a major determinant of biogeographic patterns in these groups, and we also think that this approach can be useful if it is applied to other taxa, for which we know even less. This is another study, and I will briefly explain how we have used this distance decay approach in the context of phylogenetic scaling below the species level. Okay, so it is as easy as if you have a phylogeny and you cut the tree to deeper levels each time, and you compute the distance decay of the turnover, the similarity in community compositions for each lineages, the distance decay that you can observe, and you can compare those curves. And why is that interesting? Well, this framework, we have developed this framework, and Andrés Baselga in one of his paper termed it multi-hierarchical macroecology, but it founded on the ideas that have been wonderfully explained by Belen in this book, that the processes acting at that there's some analogy between the processes acting at the genetic and at the species level, and this can be very helpful to understand what is going on in our biological community. So, in a very easy thought experiment, if you have a species and it's in a place and it's going to start expanding the range, what will we see? If it is only dispersal limited, there are only dispersal constraints, it will just spread like a water drop in all directions. Similarly, Andrés will talk a little bit later about this type of patterns that Adrián Castro Insua has also studied, one of our PhD students. We have published a very nice paper with that. But species, the range of a species can be constrained also by niche processes. So in that case, we will not expect a circle, we will expect something different, an ellipse, that the range will be deep limited. And as we are talking about large scale patterns, and the climate is structured in latitudinal bands, at this scale we'll be talking about this type of ellipses following these climatic bands. However, That's not the case at the haplotype level. If we study mitochondrial DNA, the mutual genetic parents, they are not selected. In this case, but they're within the species range. They are just limited by their ability to expand their ranges. Haplotypes, this mitochondrial nucleoplasm, you can test if they are behaving naturally or not. But if they are, we have tested in our cases, they would be just expanded, right? So we can use this expansion at the haplotype level. The patterns that we will observe at the haplotype level that would be only driven by dispersal processes with the ones that we observe at the species level that are potentially driven by dispersal processes or niche processes. So this is what we have done. So I think it's quite easy to see that the same way, if you have two sites, the same way as you compute the similarity of two communities at the species level. So how many species site 1 and site 2 share in this case? That will be only species B and C. In this example, the species are the letters and the haplotypes are the numbers. So you can do the same and compute how many haplotypes two communities have in common. So you can compute the distance decay pattern from biotic similarity at the haplotype level in the same way as you would do it at the species level. So we have done this type of assessment for two different groups. I am in the same territory, in the Iberian Peninsula, that had multiple refugia. It's supposed to have had multiple refugia during the last ice age. So what we observe is that the distance decay pattern is the same, but with different intercepts. So the fact that the decay in biotic similarity is the same at the species level and at the haplotype level is an evidence that the same processes are acting at both levels. So it points to range expansion at the species level being largely restricted by dispersal processes. So to sum up, we think it can be very useful in this context of distance decay model to compare what happens at the haplotype level and use it as a benchmark to see if we observe the same pattern at the species level, because it's not necessarily the same. And if you, with our purpose, we have done simulations to show that you do not expect same slopes when niche processes are acting. And also that the evidence of the relevance of dispersal process would come as a fractal pattern with same slopes but different intercepts. And finally, I'm going to talk about distance decay in genetic structure. And as I have said, it's not distance decay, but because we actually see the increase, but it is the same type of analysis. You have to switch it. So the relationship between genetic distance and spatial distance within each species. So this is basically what we are doing is assessing an analogous pattern. What I have just explained about distance decay in assemblages in communities, we are going to translate it to genetic distance, right? So we will expect an increase in genetic distance with spatial distance. And in this case, we are interested in the shape of this relationship. Why? Because in the theoretical framework that was proposed by Hutchinson and Templeton, they clearly explained that we would expect a linear relationship between genetic and spatial distance in the case of good dispersor. Why in the case of good dispersors, but in the case of weak dispersors, the relationship would be asymptotic, because we would observe gene flow due to dispersal when there is the increase in the curve, while when we reach the asymptotic, those are populations that are evolving independently, and it will be the result of gene drift a mutation. And if you think about places like the Iberian Peninsula with those multiple refugia where the populations have been isolated from one each other and then the expansion from them, I think it's quite easy to see how this pattern can arise. So I said that we're interested in the shape, but also with the slope, because we can compare the relative dispersal ability of taxa depending on the slope. So a flatter slope would be indicative of weaker dispersal limitation, and a steeper slope would be indicative of stronger dispersal limitation. We did this for 3 different species of Haemalacus in the Iberian Peninsula, and we saw that in every case, the asymptotic relationship was the best support model, pointing to this strong dispersal limitation. But not only that, the models were largely different in terms of slope and explain deviance, and this points to different dispersal ability among the species. So to sum up, with this study, we saw that historical dispersal limitation can be inferred from this asymptotic shape that it's analogous to how we are testing the functional shape in the distance decay models. And we also see that Haemalacus are weak dispersers, but also, and this is relevant, that very closely related species can have very different dispersal abilities. So I think it has been quite long, but I hope today have shown you some interesting approaches that you can follow if you're interested in dispersal limitation or in distance decay approaches, like how we can kind of play with these patterns from a wider range of data that you may have at hand. And I have done this in collaboration with people here at the University of Santiago and in the Natural History Museum, and I'm very grateful for everything I have learned with all of them. Thank you very much.